Okay, can somebody confirm that you're hearing me? See, I'm the only one here. That doesn't look good. Ah, good. Somebody else just showed up. For a while there, I was afraid I was on the wrong side. Can you confirm that you're hearing me?
I can hear you.
Great. Let me recording, sharing my screen now. You should see my screen. So today is August 11th, and we will be hopefully finishing microbial genetics, the lesson on microbial genetics, and maybe just starting chapter 13. There is no lab today. I'll be logging in at, I think it's 7, is that right? Sorry, in the summer, gosh, why don't I have a control of the mouse here? There we go. Yeah, I'll be logging in at 7 and I'll log out at 7:15 unless there are questions. I'll be in at 7 to 7:15 to answer questions. And that's what we'll do in the lab. If you don't have any questions, you do not have to show up in the lab. If you have questions, I'll be there to answer them. But make sure you log in between 7 and 7:15. Because once the questions are done, and once the 7:15 arrives, I'll be logging off. All right. So let me remind you, you need to get started on your unknown project. And the infectious disease paper is due this Saturday, 11:59 p.m. I think that's going to be August 13th. Yeah, there it is, August 13th. Any questions about any of that? If not, let's begin. So last time we had started talking about mutations and said that you can classify them as a base substitution, a frame shift mutation, or a macro mutation. Let's talk a little bit more about each of these. A base substitution, let me see if I can blow that up. A base substitution is when a base gets added that is incorrect. And so there should be a C over here. And in the replication of DNA, sometimes mistakes get made, not very often, but occasionally. And if the wrong base is put in there and it's kept, then that will be a mutation. And after this gets replicated, there will be no way to correct it. Before the replication, the DNA polymerase may notice that this is not pairing correctly, and it knows which base is the newer one, which meaning which strand is the newer DNA, and it will remove the newest incorrect base. You don't need to know that. I'm just telling you for your information. In which case this can be corrected, and then the C could be added. However, once this gets replicated, the T will be used as a template, and then there will be an A put opposite of it, and now this cannot be corrected. We call this a fixed mutation. Any questions about any of that? Anyways, that's the base substitution where you just change one base for what it should be. In the show there. Base substitution can cause a silent mutation, and that's where you change the nucleotide base, but you do not change the protein. And why that's the case is because if we had UUU, and this is the codons of the messenger RNA, and it would be mutated to UUC, that changes the DNA or the messenger RNA, but it does not change the amino acid. It still codes for the same amino acid. So this is a silent mutation where the DNA is changed, but the protein is not. The same amino acid is coded. So there's no effect on protein function. And that's why they call it a silent or neutral mutation. A missense mutation is where you change one base and it results in a change in the amino acid. This was actually the mutation that happened when changing the human's brown-eyed allele to the blue-eyed allele. That was a missense mutation. Such as when you have AAA, and it gets, nope, that's not changed. So I guess it's CCG, and it gets changed to TCG. That codes for a different amino acid. Instead of coding for the amino acid glycine, you code for the amino acid serine. This results in a change in the protein, and the protein probably will change its folding pattern because there's one amino acid that is different. An example of a missense mutation is sickle cell anemia. And like I said, the blue-eyed gene was a missense mutation. Any questions about any of that? All right. Another mutation is a nonsense mutation. This is a base substitution where you change the nucleotides. So instead of coding for an amino acid, I'll blow this up. So we get it changed right here. It was ACT. Is that right? I guess we're reading it down here, AUG and AAG. So it was AAG, and it's changed to UAG. This is a nonsense mutation because instead of the codon coding for an amino acid, the codon now codes for stop. Whenever you have a nonsense mutation, upstream of the nonsense mutation, you have the same protein, but downstream you have no amino acids in the protein. So it's a truncated protein. This protein likely will not function, and if it does function, it will not function at its optimal rate. Any questions about a nonsense mutation? Some examples of a nonsense mutation are some mutations causing muscular dystrophy and cystic fibrosis. All right, a frameshift mutation is where you insert or delete one or more bases. Let me blow this up. So we had AAA coding for phenylalanine, that amino acid. And now one of the nucleotides is removed, and so we only have AA there. Well, the codon is a 3 nucleotide structure, so it simply grabs the next nucleotide to read the codon. And so that will not be AAA, it'll now be AAC, coding for another amino acid. However, the next amino acid, which would be CCG, now is going to be not CCG, but CG, and then it reads the next codon A, coding for a totally different amino acid. Instead of glycine, it'll be alanine. And then once you ever have a frame shift mutation right here, the codon frame is shifted so that each codon after the frame shift will be different. And so all of the protein after the frame shift will differ. Any question about that? So if you only remove one base or add one base in the frame shift, all codons after that will differ. Any question about that? Now there is an exception, and that is if you remove 3 nucleotides or a multiple of 3, or you add 3 nucleotides or a multitude of 3. If you were to remove 3, okay, you don't have anymore the amino acid phenylalanine, so it's removed from the protein. But after that, you have the codons still coding for the same amino acids in the protein. And that's why if you have a mutation that removes a multiple of 3 or adds a multiple of 3 bases, it's not a frame shift. And I don't know what it's called, but it's a multiple of 3 bases. And you do change one amino acid. Like if you were to pull out these 3 here, you don't have the amino acid phenylalanine, but the rest of the protein is the same. And that's why it's not a frame shift mutation. Any question about any of that? Some examples of a frame shift mutation are some Tay-Sachs patients are a frame shift mutation. All right, any questions about the different types of mutations? Remember, we did mention that Down syndrome is a macromutation, where the patient has an entire extra chromosome. Normally, when a person is born with an extra chromosome, they die. But because chromosome 23 is so small, the patient can survive, but they're not normal, and we call that Down syndrome. If you are curious, the other chromosomes, which can be extra or missing and you can still survive, tend to be the X chromosome and the Y chromosome. You can have extra of those or one missing, and the person is still alive. But on something like chromosome number 1 or chromosome number 2, if you get an extra chromosome or you're missing the chromosome, it's usually lethal. All right, let's talk about the causes of mutation. We did mention that mutation can be spontaneous, where when the DNA is simply replicating itself, it makes a mistake. It doesn't happen very often. We'll talk a little bit about the frequency. It's about 1 in 10 to the million genes is a spontaneous mutation. So if you're looking at 1,000,000 genes, like in a population of 1,000,000 people, on average, you'd find one spontaneous mutation in that one gene. Of course, humans have more than one gene. Mutagens increase the risk of a mutation. So they increase the chance of a mutation. And there are two types of mutagens, radiation and chemical mutagens. With radiation, there are two types of radiation. There's ionizing radiation, which is a highly energetic radiation. When it hits an atom, it causes the loss of an electron. And then that causes the atom to become an ion because it has a positive charge. And this will change the DNA. When this molecule now bumps into DNA or whatever, it'll steal an electron and will mutate the DNA. You can also ionize the DNA and cause the mutation directly. The second type of radiation is non-ionizing radiation. And generally, there's only one type of non-ionizing radiation, and that's UV light or UV radiation. This radiation does not give the atom enough energy to lose an electron. However, UV light, when it hits DNA, it causes adjacent thymines to pair with each other. I wish I could blow this up even further. I can't seem to be able to do that. Oh, yes, I can. So the thymines, when they pair with each other, they actually become a thymine dimer. Dimer means two, and there's two thymines that pair. That's a covalent linkage between the thymines there. That prevents the thymines, when they become in a thymine dimer, it prevents them from hydrogen bonding to the A on the opposite strand of DNA. That causes a bubble in the DNA right here. And actually the cell can use that bubble in the DNA and then repair this region of the DNA. We're not going to talk about DNA repair, but it can actually repair that. The trouble with the thymine dimers, as well as a ionizing mutation from ionizing radiation, is that the DNA is altered, and now it cannot be transcribed right here. And then when it's replicated, it'll be replicated with mistake, meaning I have trouble replicating the thymine dimer correctly. And it doesn't recognize it as a thymine. All right, this other type of mutagen is a chemical mutagen. We're only going to talk about one example. Nitrous acid is a mutagen. And when this chemical gets on DNA, Let me blow this up. The nitrous acid can modify the A. It still is an A, an adenine, but it's modified by the nitrous acid. And now this A does not act like an A. And when the DNA polymerase tries to read it, it won't read it as an A, it'll actually read it as a G. And so it will put on, instead of a T here on the newly replicated DNA, it'll put on a C. And then when that happens, this newly replicated DNA cannot be corrected by DNA repair, and the mutation becomes fixed. So at this point, the cell could always repair it, and it could use the T as a template to correct it. The trouble is that it is an adenine. It's just the cell doesn't recognize it as an adenine. It's recognizing it as a G, guanine. But the cell could remove that changed A and then repair the damage. But if it doesn't and then it replicates, we now fix the mutation. And once the mutation is fixed, it's nearly impossible to revert the mutation. Okay, I'm not going to say it's not impossible, because you could have a back mutation, and that is where this will be replicated, the C will be replicated to a G. You could have a mutation where this G is mutated to an A. So that would be a back mutation. And then if that were to be replicated, you would fix it back to what it should be. So it's not impossible, but it's not very likely. The only way you could repair it would be a back mutation. And the odds of that are, at best, 1 in a million. In fact, they're less than that, because we're talking about a mutation in a gene, which would be 1 in a million. In the nucleotide, the mutation would be actually 1 in a billion, about 1 in a billion. So it's very unlikely, once the mutation becomes fixed, that it'll be corrected. All right, any question about mutagens and mutation? Here we're talking about the frequency of mutation. As I stated, the spontaneous mutation rate is 1 base is added in a mistake for every 10 to the 9th replicated bases, meaning in a billion bases that are replicated, on average, 1 of those bases will be replicated incorrectly, causing a spontaneous mutation. Now, because a gene has about 1,000 bases in the gene, that means that there's 1 spontaneous mutation in 10 to the 6 replicated genes. Any question about any of that? Mutations increase the mutation rate, and they increase the mutation rate by 10 times to 1,000 times. And that would increase the mutation rate for 1 gene in 10 to the 5th up until 1 gene in 10 to the 3rd. Any question about any of that? Identifying mutants can be done experimentally. You can test for mutants with 2 different methods. You can use positive or direct selection. And this detects the mutant cells because they grow or appear different. Like, for example, if this mutant were to walk down the street, in America, you would look at it and say, yep, there's a mutant, but it visibly looks different. And when we were looking at the blue-eyed allele versus the brown-eyed allele, that's a positive selection where there's a difference that we can detect. A negative or indirect selection detects the mutant cell because they do not grow. And I'll talk a little bit more about a negative selection and how you go about obtaining negative selection. I actually did this when I was an undergraduate, and you can test for mutations by getting a plate with isolated colonies on it, and then you get a velvet stamp. So this is a velvet cloth, and then you stamp it on the plate. That causes the cells from the colonies to move to the velvet. And then you use the velvet stamp to stamp new plates. And then cells from this colony will go there, assuming the stamp goes there. And then you do it to this one and it'll go there. Okay, so assuming that's there and there and there and there. A negative selection is where when you stamp two different media, like one containing the medium, a medium containing the amino acid histidine, and another one lacking that amino acid. And we see that in the media containing the amino acid histidine, that the colony here will grow, but on this plate, there is no growth in this position where we did stamp the cells on. So these cells are mutant and they will not grow unless you supply the amino acid histidine. And this is a negative selection for detecting mutants. And these cells are mutant right here. They will not grow on media that does not contain the amino acid histidine. Any question about any of that? All right, enough for mutation. Let's talk about genetic recombination. Recombination is the exchange of genes between two DNA molecules, where DNA from one molecule can move to another. And usually when that happens, vice versa, the DNA from the other molecule moves to the first molecule. This can happen in crossing over, where the chromosomes pair, and chromosome B is the mother's chromosome, and we're going to call it capital alleles in the gray, whereas chromosome A is the father's chromosome, and it has lowercase alleles and blue color. The two chromosomes can pair and then cross over, where the father's H and I portion of the chromosome will actually break off the father's chromosome. They changed the color here and there. Oh, well, not a big deal. And the mother's chromosome breaks off. And now we have recombination where this is part of the father's chromosome, and then that's part of the mother's chromosome. I'm going by color here, meaning blue was the father and gray was the mother, as it looks like the letters have changed from right there, which is a mistake when they made this slide, but no biggie. Now, crossing over is when two DNA molecules actually come together and pair and then break and exchange sequences. Crossing over is a little more specific than recombination. recombination is a more general term. The exchange of genes between two DNA molecules. Crossing over literally refers to the crossing of the chromosomes and then the breaking of the chromosomes where they exchange pieces. So they're similar. In all cases, the crossing over are a case of recombination, but not all cases of recombination are a case of crossing over. Any question about any of that? All right, let's talk about genetic transfer. Vertical gene transfer is when the genes from a parent move to the offspring. And we can call this transfer between generations of cells, so that it moved from the parent generation of cells to the offspring generation of cells. Horizontal gene transfer is the transfer of genes between cells of the same generation. So the genes move between a donor cell to a recipient cell. Horizontal gene transfer can happen in 3 different ways: transformation, conjugation, and transduction. Transformation is where DNA leaves one cell and then is naked DNA in the environment. And then that naked DNA is taken up by a recipient cell. So the DNA comes inside the recipient cell, and then the DNA can recombine with the chromosome, and then the new DNA can be expressed. So in this case, the DNA for A, B, C, and D moves into the cell, but only the lowercase a recombines with the chromosome and then will be expressed. This can transform the cell because this new gene can give it new properties. For example, if the lowercase a coded for antibiotic resistance, like penicillin G resistance, and then that DNA is incorporated into this cell, and then the lowercase a is incorporated into the chromosome, and then the A is expressed, this cell is now resistant to penicillin G. Whereas before, this cell was sensitive to penicillin G. Anyways, that is transformation. And this is both a process where the DNA leaves the cell, becomes naked DNA, and then gets incorporated into a new recipient cell. And then transformation is also called, you don't need to know the second term, but when the cell becomes transformed, the cell will have a new phenotype because it's expressing a new gene, the lowercase a gene. Any questions about transformation? A little picture of transformation. The cell takes up a naked piece of DNA. The cell then gets indigestion as that new piece of DNA is incorporated into its chromosome and then expressed. And when the new DNA is expressed, it then can change the phenotype of the cell so that this guy, instead of looking like this, now looks sort of, I don't know, hairy, like that. Transformation. Conjugation is also called sexual conjugation, and conjugation happens in 2 ways. In gram-negative cells, it happens via the sex pilus, and the F plasmid. We'll talk a little bit more about the F, excuse me, there it is there. The F plus cell, and then the sex pilus coming from it. The second way sexual conjugation can happen is in gram-positive cells. It can happen with a mating bridge forming. And that's where two cells come together, and then their cell membrane fuses, and then a channel gets made in the mating bridge so that the cytoplasm of this cell is now connected to the cytoplasm of that cell. And then DNA from one cell can move out of this cell and then move into the other cell. That is how sexual conjugation happens in gram-positive cells. We're going to talk a little bit more about sexual conjugation in gram-negative cells, where we have the sex pilus and the F plus plasmid. So sexual conjugation in gram-negative cells happens in an F plus cell. The F plus cell has an F plasmid in it, and the F plasmid codes for the production of the sex pilus. So this structure here coming out of that cell is encoded by the plasmid, the F plasmid, in this cell. The sex pilus, let me go back. The sex pilus that comes out of this cell literally skewers an F minus cell, which is this cell, and then acts as a winch to bring these two cells together. Now they don't have to be this close because the sex pilus is hollow, and the F plasmid from this cell can move through the sex pilus to enter the F minus cell. Any question about any of that?
I had a question.
Uh-huh.
So during the sex pilus conjugation or whatever, they're only passing on the gene to form a sex pilus. That's the only gene that's being passed on.
Not exactly. That's just the ones we'll be talking about.
Oh, okay, gotcha.
Because
So other
the
genes can be
plasmid-
passed on that way?
Yeah, the plasmid, the F plasmid, which is the F plus factor, the F plasmid from this cell will make a copy of itself. I haven't talked about that yet. So let me go on down here. When the plasmid makes the sex pilus, which will skewer this cell and then can bring them right next to each other. They don't have to be right next to each other because the sex pilus is hollow. And then the F plasmid will replicate itself. So there will be two copies. And then one copy of the F plasmid will move through the sex pilus and into the F minus cell. Okay. And this is usually what happens, but occasionally more than the F plasmid may move into this cell. Okay. And that's all I want to say, that occasionally you can get more DNA moving from this cell into that cell. But we're only going to talk about the F plasmid moving. Any question about any of that? All right. Now let's look at this F minus cell. It's called F minus because it doesn't have the F plasmid. It doesn't make the sex pilus. And if you look here, there's a few other differences too between the F minus cell and the F plus cell. The F plus cell is a little longer. It's a little rounder. The F plus cell has a sex pilus, And we already mentioned the sex pilus is encoded by the genes on the F plasmid. And then this cell is a little bit more hairy, meaning you have the structure is coming out of it. And the F minus cell is more round and less hairy. Well, when the F plasmid moves from the F plus cell to the F minus, we now have this cell with the F plasmid. And this F plasmid codes for the sex pilus. So this cell will grow a sex pilus. So the two of them will have sex piluses. I guess that would be sex pili. And that changes the phenotype of this cell from F minus to F plus, because this cell will have the F plasmid, it will grow a sex pilus, and it will change into the F plus cell. And that includes the fact that this cell, once it grows the sex pilus, can engage in recombination or conjugation with an F minus cell. And of course, that cell can too. Any question about sexual conjugation? All right. If you're wondering why it's called sexual conjugation, ask your significant other why it's called sexual conjugation when the DNA moves from one cell into the other cell. All right. The third way that genes can move between cells of the same generation is by transduction. Transduction involves a virus, and we haven't talked about viruses yet, so let me talk a little bit about viruses, and then you'll understand it maybe a little better after we've talked about viruses. But we have, in generalized transduction, which is all we're going to talk about, a cell that will be infected with a virus. And what the bacteria virus does is it attaches to the cell and then pumps its viral DNA into the cell. And because the virus wants to call the shots in the cell, one of the first genes expressed on the viral DNA is to code for the destruction of the host cell's chromosome. So this gene codes for an enzyme, and what it does is it cuts the host cell's chromosome. Why it does that is that the viral DNA wants to call the shots in the cell, and an easy way of calling all the shots in the cell are to degrade those chromosomes. And now the only DNA controlling the cell are the viral DNA. Any question about any of that? All right. What viruses do when they infect a cell, at least lytic viruses, is they will then replicate their genome inside the cell, and then the genome will be expressed, making messenger RNA, which will then make the protein for the capsid of the virus, meaning the viral protein. The viral protein and the viral DNA will then get together. And then the last thing the viral DNA codes for is the lysis of the cell, and that will release the newly replicated viruses. Not working, there we go. But when viruses replicate, they sometimes make a mistake. Let's take a look at this virus here. You'll note that all of these viruses, these 4 here, have incorporated the viral DNA because it's a purple color, or reddish purple, I should say. But you see this virus, the protein portion of the virus, got a piece of blue DNA, and the blue DNA is a piece of the host cell's chromosome. Why viruses make mistakes is they make lots of offspring, and if they make one offspring that isn't correct, they're not worried about it because they make lots of offspring. Okay, but now let's see what happens when this virus infects a cell. It will infect the cell attached to it and then pump its genome into the cell. However, what is pumping into the cell is not viral DNA. It's actually DNA of this chromosome to this cell. This DNA can then recombine with the host cell's chromosome, and then that DNA can be expressed. This can bring this cell new genes. And this cell, let's say it's sensitive to the antibiotic tetracycline, and this cell is not. Right there, we have the gene for tetracycline resistance. And this piece of DNA is incorporated into this virus. This virus then can move DNA from this cell into this cell, assuming that this virus attaches to this cell and then dumps in its DNA. That DNA from the virus can then recombine with the host cell's chromosome, and then this DNA from the virus can be expressed. When it's not really from the virus, this DNA is from the chromosome of this cell. This can bring this cell new genes, such as tetracycline resistance. This cell never had that before incorporating the DNA from this virus. Any question about transduction? All right, so let me remind you that DNA can move from cells of the same generation, trying to get there, by 3 different means. They can get there by transformation, by conjugation, and transduction. Transformation involves the DNA leaving from one cell, and that cell is probably going to die. And then the DNA is naked DNA in the environment, and that DNA can be incorporated into a recipient cell. Conjugation, we only talked about it with the gram-negative cells, but that's all we're going to talk about. And the DNA can move from one cell into another cell. And this happens by the sex pilus and the F plasmid. The F plasmid can move from one cell to another cell. If the F plasmid were to incorporate a gene like tetracycline resistance, it would then pass on that gene to the new cell, the recipient cell, and then both cells can become tetracycline resistance. Any question about any of that? All right, and the third way that horizontal gene transfer can happen is by transduction. Transduction involves a virus, and then viral multiplication makes a mistake. And there can be a few viruses, or maybe only one, that do not have the viral genome. It has the genome, or a portion of the genome, I should say, of the host cell's chromosome. All right. If there's no questions, let's talk a little bit about plasmids. Plasmids are small, circular, extrachromosomal DNA. They do contain genes, but how a chromosome differs from a plasmid is a plasmid is very short. It only has a couple of genes on it, like 1, 2, 3, 4, 5, 6, maybe 7 genes on it. So only a handful of genes, plasmid. The bacterial chromosome, on the other hand, has something like 20,000 genes, depending on which bacteria it is. And so it's much larger, has many more genes. They both are made up of DNA and they both contain genes, but the plasmid is different just because it's floating around in the cytoplasm. It's much smaller than the bacterial chromosome, which most of the time in a prokaryotic cell, the bacterial chromosome is floating around in the bacterial cytoplasm. Plasmids can replicate independently of the cell's chromosome. And we actually saw that up here, where this plasmid replicated, not because the chromosome was replicated, but because the F plus cell had found a F minus cell and then skewered it and brought the two cells closer together. What that F plasmid did was it replicated so that it could send one copy into the F minus cell. The bacterial chromosome is not replicating here. Showing you independent replication, which is the way most plasmids replicate. All right, you should know there are different categories of plasmid. You do not need to know what the names of these categories are or anything about these categories, okay? All right, any question about plasmids? If not, let's move on to transposons and insertion sequences. A transposon is a piece of DNA that can move from one region of DNA to another. Did you catch that? A transposon is DNA that moves around from one place to another, meaning the DNA moves. Transposons always contain an insertion sequence at the end, and that's inverted repeats that helps the DNA ligate out from where it's moving from, and then it helps the DNA ligate or cut where the DNA, the transposon wants to move to. Any question about any of that? All right. Transposons, besides having the inverted repeat or the insertion sequences, have between the inverted repeats, they always have the transposase gene. So to be a transposon, you have to have the enzyme transposase. And what this enzyme does is it cuts the DNA where this transposon is, and then it will anneal the DNA back together. And then the transposon cuts the DNA where the transposon is going to insert into. And all transposons have these properties. However, complex transposons carry other genes besides transposase. Complex transposons usually have I don't know, transposon on this side of the DNA and on this side of the DNA. And then they have the extra DNA in the transposon in between them. If transposons contain extra DNA, they can move the DNA from one location in the cell to another, such as kanamycin resistance gene. Transposons can also move into a plasmid, like from the chromosome, they can move this piece of DNA onto a plasmid. And then because plasmids are more likely to move between cells, the transposon can move into a new cell via the plasmid. All right. Any question about transposons? I guess what I want to say is transposons usually do not give a cell a new gene because a transposon just moves from one region of DNA to another region. However, that shuffles up the genes and makes new combinations possible. But recombination also does that. Did I talk about that? And that is, recombination does not I never talked about that, so let me back up and talk about that. Recombination does not give the cell new genes, but it puts alleles next to each other that used to not be next to each other, such as the lowercase g and the uppercase h can now be put together by recombination, whereas before the two weren't together. And that shuffles up the DNA in a cell, mixing it up. Recombination mixes the DNA, and then transposons also mix up the DNA. Oops, that's the wrong one. Here we go. And then if transposons move from the chromosome to a plasmid, that can encourage the newly moved DNA to move to another cell by horizontal gene transfer. So transposons and recombination do not give the cell new genes, but they shuffle up the genes, making new combinations possible. How the cell gets new genes is by transformation, conjugation, transduction. As long as the gene exists in a population, transformation, conjunction, or transduction can move the gene from one cell to another so that the recipient cell receives new DNA. And of course, the other way a recipient cell can receive new DNA is by mutation. If the gene does not exist in the species or in the population, the only way you can get it is by mutation. And as I mentioned, the human genome over 10,000 years ago only had one gene for eye color, and that was the brown-eyed allele. 10,000 years ago, we got a mutation giving us another gene, and that was the blue-eyed allele. Any question about any of that?
I did have a question.
Uh-huh.
What would be the point Or like the reason behind just shuffling the genes around? Does it just change like gene expression?
Yeah, it's to make new combinations possible. For cells, it's kind of hard to talk about, but let's talk about children, for example. Where's recombination? Two children that are born from parents, they may look similar because they have similar genes, but unless they're identical twins or triplets or something like that, they will not look the same. And that's largely because they have different genes, and recombination shuffles up those genes to make more combinations possible.
Got it. I
It
was
is
just trying to think
important.
of like a practical reasoning behind why that would occur, but that totally makes sense.
Why is just because to make there being more genetic variation in the population.
Got it. Okay, thank you.
And that's good for the population. It may not be good for the individual. It may be neutral. Like I said, but it's good for the population because it makes more individuals with different genetic variation. So there are differences among the individuals within the population. And that's
Got
good
it.
for the population because if the environment changes, then the majority or maybe all of the individuals in the population would have a hard time living in the changed environment. But because of recombination, there are all these different combinations of genes which are now possible. And so there will be a few individuals that can do very well in the new environment. Do you get that?
Yeah, that totally makes sense. Thank you.
Okay. Like with the polar bears, they're having trouble living in a new world because of global warming. The environment is warming. And so they're having to change their lifestyle. If you didn't know, a polar bear, what it used to do was it would find a seal hole, which would be where the seals would come up to breathe. And there'd be so much ice, the seals would make this hole and they'd come up to it to breathe. And the polar bears just came to this seal hole and then waited for the seal to breathe, come up to breathe, and then the polar bear would catch him and get an easy lunch. Well, now because of global warming, the ice has melted and the seals can come up nearly any place to breathe. They don't have to come to a seal hole. And so the polar bears are having a hard time surviving with global warming. And their population probably isn't big enough so that they have enough genetic variation to survive a changing environment. We'll see what happens, but my guess is they will probably become extinct because they're not doing too well in the warming environment. And recombination, like I said, it's just good for the population. All right, let's move to the last slide here. Genetic variation, at the very first, if there is no genetic variation, the only way it can be created is by mutation. Once it is existing in the world or in the species, it can now move among individuals by transformation, conjugation, and transduction. And it can also be brought about in individuals by mutation. Crossing over and transposition, just make new alleles possible so that you can get new combinations within the individuals. All of this gives rise to genetic variation in the population. And then that genetic variation will drive evolution, which will happen by natural selection. Meaning natural selection determines which way the species will evolve. Like for polar bears, natural selection will be selecting for those polar bears which have another way of hunting with global warming. And if there's enough genetic variation, the polar bears will survive. My fear is the polar bear population isn't very big, and so they may go extinct. But anyways, genetic variation resulting from mutation, spread around by crossing over and transposition, meaning the genetic variation can make new gene combinations with crossing over and transposition. Then transformation, conjugation, and transduction can move the new genetic variation to new individuals, and that drives evolution. All right, any question about that? Okay, that's it for genes and evolution. Any questions about chapter 8? If not, let's close that down. I think I took the 2 here out, so I'll save that. And let's begin chapter 13. Chapter 13 are viruses, viroids, and prions. The major goals and the rough outline are shown in this slide. So know the terms. What's a capsid to a virus? What's an envelope? What's a spike? What's a bacteriophage? What is a lytic life cycle? What's the lysogenic life cycle? What's a prophage? What's a provirus? What is the latent life cycle? What is a persistent infection? What is a prion? And what is a viroid? Understand the viral characteristics, what their structure is, and the classification of viruses. Also understand about viral replication, viral multiplication. And then lastly, we'll talk about prions and viroids. Any question about what we're doing? All right. Viruses only have one goal, and their goal is to replicate. How do they do this? They invade their host cell, hijack it, and then force the host cell to make viral copies, which then escape from the host cell. Let's watch a little movie about that, if I can get it. Little video. I probably don't need this.
the microscopic world of bacteria, a miniature act of piracy is about to take place. T4 bacteriophages. With their spider-like tail fibers and prism-shaped heads, these viral marauders plan to hijack an unsuspecting bacterium and use it as their own metabolic nursery. This E. coli bacterium has seen its last day. Binding to its surface, the virus injects its precious cargo of genetic material into the cell, commanding it to produce a new batch of T4 bacteriophages. In a final act, the bacterium is forced to self-destruct, releasing hundreds of viruses ready to kill again.
Okay, any questions about that video? Well, that sounded rather loud. Some general characteristic of viruses. They are inert when they are not within their host cell. In fact, they display little or no metabolic activity when they are not inside their host cell. So a virus outside of its host cell has no metabolic activity or little metabolic activity. It only has metabolism when it is inside its host cell. A virus requires a living host cell to multiply. So we can say a virus is an obligate intracellular parasite. It requires the host to replicate. Viruses multiply within a living cell by using the host cell's enzymes, nucleic acid, amino acids, and ATP. In fact, viruses have no ATP generating mechanism. They simply use the ATP that is in their host cell. Viruses contain DNA or RNA as their innermost molecule, which is their genome, but they never contain both. So viruses have a genome either of DNA or RNA, but never both. Now, if you remember in cells, we only have a genome of DNA. Viruses can have a genome of DNA or RNA. And the genome is the innermost molecule of the virus. So here we have a virus with this side cut away. Look inside, and the purple or blue is the viral genome. The viral genome is then surrounded by a protein coat, shown here. And then some viruses have the protein coat inside an outer lipid envelope, which will go around the virus, the viral protein coat, I guess I should say. Not all viruses have an outer lipid envelope. Some of them do not. Some of them do, some of them do not. The host range of a virus are the cells that the virus can infect, and these are called the host cells of the virus. The host range is determined by specific host attachment sites, like cell walls or flagella, certain molecules on the host cell that we call receptors. And the host range is also determined by certain cellular factors necessary for viral multiplication. I can't think of what that would be, but if you had a cell with no ATP, the virus would not replicate because it needs ATP that's, that is supplied by the host cell. Most viruses infect only cells of one species or a few species. And their binding is very specific. There will be specific attachment molecules on the virus that specifically bind to receptors on the host cell. Any question about that? So most viruses can infect only one or very few host species. There are some exceptions, but most can only infect one or very few species. Like HIV can only infect human cells and then one chimpanzee. So very specific. And in fact, it doesn't infect all human cells. I don't know how many human cells it can infect, but it doesn't infect, for example, your nose cell or your skin cell. I don't think it infects your brain cell, not your brain cell, your bone cell, but it can infect your T cells. And I think it infects your macrophages. And it infects a few other cells of humans. Okay, but it does not infect all human cells. And that host range is due to the high specificity of the attachment molecules on the virus and the receptors on the host cell. Any question about any of that? All right. A better example than HIV is, now I lost the name of that. What is that disease called? I just had it on the tip of my tongue. That would have been a better example because only humans can be infected. Polio. Polio virus can only infect human cells, and it tends to infect nerve cells. Okay, meaning it doesn't infect most other cells. Viruses are small. You don't need to know the size. Let me see if I can blow this up. The smallest virus that you've ever heard of is the polio virus, but there are smaller viruses. But I'm going to guess no one has ever heard of bacteriophage MS2. Okay, so poliovirus is about the only smallest virus you've ever heard of. Viruses range in size from about 20 nanometers to 440 nanometers in diameter. If you go with their length, it's 20 nanometers long to nearly, they're a little less than that, but nearly 1,000 nanometers long. That would be 1 micrometer long. And that's Ebola, the very longest virus. But it's not the biggest virus because it's so narrow. The biggest virus we're going to talk about is megavirus, which is about 0.44 micrometers in diameter. The largest virus approaches the size of the smallest prokaryotic cell. So about the smallest prokaryotic cell is about 0.44 micrometers in diameter. Come on, mouse. And that's the largest virus. Viruses, though, on average are much smaller than prokaryotes. And I'm going to say they're about 10-fold smaller than a prokaryotic cell. And then I stated prokaryotic cells are about 10 times smaller than your average eukaryotic cell. So here we have a red blood cell, which is actually a small eukaryotic cell. And then E. coli, which is a little bit larger than your average prokaryotic cell, because it's a little bit longer. And then different viruses. Any question about the virus relative size? When we're talking about viruses, it's important to talk about the viral structure. Viruses have different structures, like they have a genome, and then all viruses besides having a genome, which can be either DNA or RNA, they have a protein coat that we call the capsid, and all viruses have that. Some viruses have, in addition to that, they have the structure of an envelope around the protein coat. For viral genomes, the genome is either DNA or RNA, but it is never both. Viruses have double-stranded DNA as their genome, which is normal. That's what our genomes are. But some viruses have single-stranded DNA. And that can be seen in cell DNA. Whenever the cell is replicating its DNA or transcribing the DNA, we have single-stranded DNA, at least at the site of replication or transcription. Okay. Viruses can have double-stranded RNA genomes. This is unheard of in cells. Cells do not have double-stranded RNA. Okay. Viruses can also have single-stranded RNA molecules for their genome. If we're talking about a single-stranded RNA molecule, it's important to distinguish, is it a positive strand, single-strand RNA, or a negative strand, single-strand RNA? The positive strand, single-strand RNA molecule can be read by the ribosomes. And that would be something like AUG. The ribosome can read it and then translate it into protein. The single-strand RNA genome that is a negative single strand cannot be read by the ribosome. And it would be the corollary to the positive strand. So instead of having AUG, you'd have UAC. Any question about any of that? For the nucleic acid, we can have linear genomes of a viral genome, which is normal. Our cells are, well, normal for eukaryotes. Our genomes are linear. And then viruses can be circular genome, which that is similar to prokaryote DNA, prokaryote genomes. They're circular. A circular genome, remember, it's in a circle, and that is it will begin where it will end. Any question about any of that? Some viruses, just a few, have their genome in several segments, like the influenza virus has their genome in several segments or several pieces. This should not be surprising to you because humans have their genome in several pieces, and we call each piece a chromosome. Okay. Any question about any of that? All right, let's talk about the capsid. This is the structure surrounding the viral nucleic acid. The capsid, let me blow this up. It's made up of capsomeres. And so all of the protein, which is, I don't know, a pinkish color here, is the capsid. But it's made up of capsomeres, and each of this little round structure in the capsid is a capsomere. In this virus, there's only one capsomere in the capsid, and it's just repeated. That doesn't have to be the case. Let me skip many slides. Here we go, right here. It's not too far. Shrink this down. Here we have different capsomeres in the head portion of the virus and different capsomeres in the sheath and different capsomeres in the tail fiber and different capsomere, excuse me, there's the tail fiber, different capsomeres in the base plate. And these are just different proteins. Any question about any of that? So in this protein code of the virus, there's only one protein, and it's just repeated to make the protein code. Some viruses have a lipid envelope surrounding the protein code. Let me see if I can blow this up. So there we have the envelope. And that would be this blue and purple region here. If you look inside, you can see the capsomere, the capsid right there. And then there is the genome. Okay. If the virus has an envelope, it usually has spikes which protrude from the envelope. And those would be those reddish purple things coming out of the envelope. The envelopes usually serve as antigens for a host that is infected with the virus. So for example, when we make antibodies to a virus, the antigens typically are the molecules furthest out on the virus. And on this virus, the spikes are the molecules furthest out. So on this virus, the spikes serve as the antigens. Any question about that? Now for the virus, although the spikes can serve as an antigen to a host, meaning that's what the host recognizes, but the spikes typically serve as an attachment point for binding to the host cell. And the reason for that, let me back up, is once again, the spikes are the furthest molecule out on the virus. And it's usually the furthest molecule out that serves as the attachment molecules for this virus. So the spikes are serving two things: as an antigen for like humans, the host, and for the virus, they're serving as attachment sites. These have the attachment molecules for attaching to our receptors. Now the spikes are usually glycoproteins, meaning they're more than just proteins. They're usually glycoproteins. All right, any question about viral structure so far?
I just had a question about the PowerPoint. It says if no envelope, it is and I was curious what that was.
I didn't see that.
On slide
Hey,
7,
let me back up. Where is that?
on slide 7 under capsid, the last little bullet point said
Ah,
if no
yeah.
envelope,
I haven't
it's
told you, if the virus has an envelope, we call it an enveloped virus. If the virus has no envelope, it is called a naked virus. So if the virus is like this, only has the genome and its capsid, it's a naked virus. This is an enveloped virus. Thank you for asking that, because I meant to say that and I forgot. Didn't see
no
it.
problem. Thank you.
All right, let's talk a little bit about the general morphology of viruses. The morphology is the shape of the viruses. Viruses are usually classified on the basis of their capsid structure. If the capsid is helical, we call them helical viruses. If the capsid is a polyhedral surface, then we call it a polyhedral virus. Remember, a polyhedral virus is one that sort of looks like a many-sided dice. And there we have an actual polyhedral virus, although there's spikes protruding away from the polyhedron. But the capsid determines the helical shape. For example, on Ebola right here, this does not look like a helix to me, but Ebola has more than just a capsid, meaning it's envelope. And we can't see the capsid, at least in this picture. Apparently, it's helical, the capsid. The third way, so viruses are normally called helical, polyhedral, and the third one is complex. A complex virus can take on essentially any shape. Like this is a complex virus, this is a complex virus, and so is this. If you recognize this shape, that's from bacteriophage T4, it's taking on the shape of the lunar lander that landed on the moon in the Apollo missions. Any question about any of that? We'll talk a little bit more about bacteriophage T4 because it is the virus that has been studied the most. So we know more about this virus, bacteriophage T4, than we do about any other virus. And the main reason why T4, bacteriophage T4, was studied so much is simply because it grows in E. coli. And so it was therefore an easy virus to study because most microbiology labs already had E. coli. T4 begins to the T even family of viruses, and they all have this same shape, like T2, T4, and T6 are in the T even family. T4 is a virus which infects E. coli, so it is a bacterial virus. Any question about that part? If not, I want to talk a little bit about viral infection and then the host immune response. If a virus infects an animal, and then the virus will infect the animal cell to replicate in the animal cell, which can stimulate the host, the animal, to produce antibodies. And what happens is the host recognizes the virus from its antigens and then makes antibodies against those antigens. And the antibodies recognize and bind to the antigens of the virus, which will be the surface proteins of the virus. Any question about any of that? That's actually important to us so that our immune system can respond to a viral infection and then mount an immune response against it. And that is how we eliminate most viruses. Okay, even polio was eliminated by that way. And then that's how we get rid of the human cold when we have it. We mount an immune response against it. Some viruses are harder to fight off, and that's because the viral protein can change or mutate frequently. Therefore, different viral strains can infect the same individual more than once. Can anyone give me an example of that? Where more than one viral strain can infect the patient more than once.
COVID.
Yeah, COVID-19 is a good example, where the virus changes, mutating, and then the virus can be different, and our immune response will no longer at least respond well, and so you can be infected a second time. And I know people have been infected at least 3 times. I imagine there's been some even 4 times, but I don't know of any cases. Another example of a virus that changes very frequently, even more than COVID, is influenza virus. And because the virus changes so often, once you get the flu shot to immunize yourself against the flu, that flu shot will not be any good after 3 years. Okay, and we'll talk a little bit about the flu later in the term. We'll talk about COVID later in the term too. I'm not sure why COVID is changing so rapidly. Why it mutates so rapidly. With the flu, I do know why it's changing so rapidly, and we'll talk a little bit about that. All right, viruses are grouped into families. The families are based on their type of nucleic acid, their replication strategy, their morphology. Family names end in viridae. The genus name ends in virus. So like herpes viridae is the family of herpes viruses. And then the genus would be herpes virus. The virus has a species, which is a group of viruses sharing the same genetic information and the same ecological niche, meaning they'll infect the same host. You'll like this though, and that is when we name viruses, viruses only have common English names, like the polio virus. That is a common English name. There is no Latin naming system for viruses. And apparently all of the early virologists were English speaking, and they gave viruses their English names. And then the non-English speakers started studying viruses. And then lucky for us, they decided to keep all of the English names. And so viruses have simple common English names like polio virus. And that's all there is. Viruses also have subspecies, and they are designated by a number. A subspecies in the human population would be a race. Like the blacks are a race of humans, and then the European whites are a race, a subspecies of the humans. Okay, so that's what we mean by a subspecies. We will simply call the subspecies by the name, like poliovirus, and then there will be a number like poliovirus 1 and poliovirus 2. Now, the subspecies are very important in the virus because of the effect the virus has on people. For example, one subspecies of the poliovirus gives humans the polio disease. All right, and that's not surprising. What is a little surprising is another subspecies of the poliovirus we use as a vaccine to prevent humans from getting the disease polio, meaning one subspecies is a live virus that we use to vaccinate people against getting polio. And that is the Sabin vaccine. It's just a subspecies of the polio virus. Any question about that? That tells you how different the subspecies can be. One gives us the disease, the other one we use as a vaccine to prevent getting the disease. And this tells you that most viruses do not change rapidly because the Sabin vaccine was developed in the 1950s and it is still good today, meaning the polio virus, we made the vaccine in the 1950s and we're still using it today. What is that? Over 70 years. Or about 70 years, because I don't know when in the 1950s it was made. Okay, and the virus hasn't mutated. We can still use the vaccine. Too bad COVID-19 isn't that way. COVID-19 mutates much more frequently, and they expect that we will have to get a vaccine about every 6 months. They don't know exactly how long it'll be. It might be, if we're lucky, every year. But so far, it looks like it's going to be about every 6 months. You'll need to get a vaccine. And if you don't know, the current COVID-19 vaccine no longer prevents you from getting infected from the variants which are circulating now in the human population. And I'm trying to remember the name of the virus. It's something like BA4 is the variant which is circulating now. And the vaccine does not prevent you from getting COVID-19. However, they believe it prevents you from getting serious COVID disease, and therefore it's helping prevent deaths. All right, any question about any of that? If not, let me go ahead and talk about this slide and then we'll end here. A viral taxonomy, we already mentioned this. The family is Viridae, so with the herpes viruses, the family name is Herpes viridae. The genus name is simplex virus or herpes simplex virus. The common name, human herpes virus which actually isn't being used, and we'll call it herpes simplex virus. The subspecies, herpes simplex 1, and that's viral taxonomy. Any question about any of that? All right, if not, I'm going to end here. If you have any questions about the lab, I will be there from 7 o'clock to 7:15. You do not have to show up in the lab if you do not have questions. And you don't want to hear anybody else's questions. Okay? All right. I will see you later unless there's a question. All right, goodbye.
Thank you.
You're welcome.